Hello everyone, welcome back to my YouTube channel. So today our topic is hemolysis and fragility in red blood cells. So first we will see the definition of hemolysis. So what is hemolysis? Hemolysis is destruction of formed element. To define more specifically, it is process which involves the breakdown of red blood cells. Okay. Now we will see definition of fragility. So what is fragility? Fragility is susceptibility of red blood cells to hemolysis or tendency to break easily is called as fragility. Fragile means easily broken. Now we will see types of fragility. There are two types of fragility. First is osmotic fragility, which occurs due to exposure to hypotonic saline. We will see the example of this. And second is mechanical fragility, which occurs due to mechanical trauma or any wound or injury. Under normal condition, only old red blood cells are destroyed in RE system. In abnormal hemolysis, it is process by which even younger RBC are destroyed in large number by the presence of hemolytic agents. Now we will see the process of hemolysis. Normally plasma and red blood cells are in osmotic equilibrium. When the osmotic equilibrium is disturbed, the cells are affected. For example, when the red blood cells are immersed in hypotonic saline, the cells swell and rupture by bursting because of endosmosis. The hemoglobin is released from the ruptured red blood cells. Now we will see the fragility test. So in fragility test, it is test that measures the resistance of erythrocyte in hypotonic saline. It is done by using sodium chloride concentration at different levels from 1.2 to 0.2. We can see in this diagram that sodium chloride concentration in every tube is different. This tube is called as Con's tubes. So we can see 0.7, 0.65, 0.60, 0.55. The solution at different concentration are taken in series of Con's tubes. So after taking sodium chloride, then 1 drop of blood to be tested is added to each tube. The sodium chloride solution and the blood in each tube are mixed well and left undisturbed for some time. Results can be analyzed by observing the tubes directly or by centrifuging the tubes after 15 minutes. After 15 minutes, we can see the results. So first we will see the direct observations. So first is if there is no hemolysis, fluid in the tube appears turbid. Second, if hemolysis is started, turbidity is reduced. Third, if hemolysis is completed, the fluid becomes clear. So this is 3 direct observations. After this we will see observation after centrifugation. So first is if there is no hemolysis, cells sediment at the bottom with clear colorless fluid above, that is plasma. Second, if hemolysis is started, cell sedimentation is less and fluid become slightly reddish because of release of small amount of hemoglobin from few hemolyzed red blood cells. Third is if hemolysis is complete, fluid becomes more reddish without any sedimentation due to release of more hemoglobin from all the hemolyzed cells. Now we will see the fragility index for fragility after 20 minutes. No hemolysis up to 0.6%, onset of hemolysis around 0.45% and completion of hemolysis around 0.35%. At 0.45%, only older cells are destroyed because their membrane is very fragile. So these cells cannot withstand this hypotonicity. But younger cells are not affected. At 0.35, even the younger cells are destroyed. So now we will see condition when hemolysis occur. First is hemolytic jaundice. Second is antigen antibody reaction. Third is poisoning by chemicals or toxins. Fourth is while using artificial kidney for hemodialysis or heart-lung machine during cardiac surgeries. But this is very rare occasions. Now we will see what is hemolysis or hemolysis agent. So hemolysis or hemolytic agents are the substance which cause destruction of red blood cells. The hemolysis are of two types. First, chemical substance. Second, substance of bacterial origin or substance found in body. Now we will see chemical agents or chemical substance that causes hemolysis and called as hemolytic agent. First, alcohol. Second, benzene. Third, chloroform. Fourth, ether. Fifth, acids. Sixth, alkalis. Seventh, bile salt. And eighth is saponin. Ninth is chemical poisonings like first, arsenial preparation. Second, carbolic acid, nitrobenzene, and last, resin. This was chemical substance or chemical agents. Second, we will see substance of bacterial origin or substance found in body. First, toxin substance or toxins from bacteria. From bacteria like Streptococcus, Staphylococcus, Tetanus, Bacilli. Second, venom of poisonous snakes like cobra. Third, hemolysis from normal tissues. So here we completed our today's topic called hemolysis and fragility in red blood cells. Thank you for watching. Please like and subscribe.